Okay, we're going to get started. Session 2, we're going to look at data sharing models. I'm going to speak very loudly so perhaps the people in the hallway can come and sit down. Design, best practices, and lessons learned. Our chair for this session is Jeff Nye. Jeff is a vice president and global head of neuroscience external innovation at Janssen Pharmaceutical Companies of Johnson & Johnson, essentially responsible for the strategy and implementation of external R&D in the neurosciences. So, Jeff, take it away.
Thank you very much. It's an honor and a privilege to participate in this workshop and to have the opportunity to work with such a fine and distinguished panel of organizers and also of speakers. As you've heard today, and I hope you're as inspired as I have been, a key principle that we heard about was that clinical data is a public good. And we heard that from several of the speakers, and I thank them for expressing their vision. It affects many stakeholders, and we began to see about all of those stakeholders. The patients, subjects, participants are a key group, and we heard about that in the discussion. The researchers from academics who have a different vested stake in it, but their point of view is, of course, of considerable interest because they need to be motivated culturally and otherwise, as well as pharmaceutical companies, and we talked about people who are doing tertiary analyses or meta-analyses of the study data as well, and citizen scientists who may be eventually perusing databases on the web and learning new things that could be of some importance. So this is really a common interest with multiple stakeholders. We heard about companies moving forward to advance more transparency and openness, and the issues around public trust of companies, which are important issues that I think will come out and be important. I want to emphasize two parties that were sort of not mentioned. One is the regulatory view, which I think is an important consideration, recognizing that the regulators have an objective to provide for the public health and to guide and advise companies as they develop new products, and as well as to regulate clinical trials done by anyone. And also economic development, and a quotation from Patrick Kennedy, who we work with in the One Mind for Research campaign, is that there's no better economic development than to find solutions for healthcare problems. So it becomes a stake of the entire economic system to recognize and to improve on clinical medicine so that we don't waste money on bad treatments, so that we don't have patients having devastating outcomes from untreated illnesses. So I want to thank the prior speakers for their excellent vision, which I think we all heard and significantly appreciated. This session is an attempt to dig more deeply into all of the issues that I think were raised in the prior session. We're going to talk about some existing data sharing models from two perspectives. One is those that provide more access and visibility to data, which is somewhat distinct than those that provide pooling and analysis of the data. So we've divided this roughly up into two parts for this next session so that we can focus on it. The third part then will be a series of brief presentations where we can really get into the analysis of some of the true challenges from the legal perspective, from the methodology perspective, and to go deeper into the cultural perspective. And then we have a moderated discussion from Sally Oken. So moving forward, first I'd like to introduce Deborah Zarin, who you already heard about, is the director of the clinicaltrials.gov initiative, and I think she was introduced. So without further ado, let me introduce her now.
Thank you very much. Oops, I think I need my slide. Okay, got it. First, a disclaimer, because it'll be easy to misunderstand. I'm an employee of NIH. I'm the director of clinicaltrials.gov, as you heard, but my comments are my own. They're not official NIH policy by any means. And in particular, since there have been rumors in the past, I want to be very clear. Clinicaltrials.gov does not require patient-level data. We do not accept, I should call it, participant-level data, and there is no future in which we will be accepting participant level data. So if you hear those rumors, they've been very hard to put out in the past, so let me try to prevent some. Okay, I'm just going to breeze through things that we've basically already know and you've already heard, but it's really important to go back because the focus of this talk is sort of why are we worried about summary level data, or why isn't that enough, and what have we learned from clinicaltrials.gov. So why do we do clinical trials? This says we have two options, either an evidence-based treatment or an exciting, risky alternative. So this came from my days when people seemed to be really into innovative treatment, whether or not they were good or bad. So you all know evidence-based medicine, and the main point here is the bottom bullet. Most decision makers depend on summary data from journal articles. Okay, the decision makers at FDA are relatively unique in that they routinely have participant level data, but the rest of us, either clinical or policy decision makers, are really left with summary data. So there are three key problems that interfere with the practice of evidence-based medicine. You all know this, not all trials are published. When they're published, they don't always include all the pre-specified outcome measures, which leaves a lot of room for cherry picking. And unacknowledged changes can be made to the protocol that would affect the interpretation of the findings if the reader knew about them. And so example of that is changes to pre-specified outcome measures. All of these contributed to the development of clinicaltrials.gov, which was established in 2000. People register trials at inception, in theory. Registration contains key protocol details. We now have over 130,000 trials listed from around the world. The results database was added in 2008 as a result of federal legislation, and you enter results after the trial is completed. We have summary results, let me emphasize that again, not participant level results, and we now have results for over 7,000 trials, and mostly what I'm talking about is our experience with that. So you saw this earlier when I presented it, but again, just to emphasize that we're talking about the analyzed summary results, but also we've gained insights into how you go from that uncoded data, that first step that Dr. Loader showed some examples of, down to the summary results. So again, decision makers depend on summary results. And why? Well, they're convenient. It's way more convenient. As you know, from my former days as a clinician, it was easier to read a journal article with some summary data than to go through individual participant level data. That would be crazy. And we assume that they're an accurate reflection of the underlying participant level data. And we assume, therefore, that there's little room for subjectivity. I think some holes have already been punched in that this morning, but that's basically the assumption that society, our world, has been working on. So just to emphasize again, levels of transparency, clinicaltrials.gov is over on the left, which is to provide evidence that the trial exists. There didn't used to be evidence that trials existed, and a summary of the protocol details. We don't currently provide access to the full protocol, and then the results database is summary results, and this conference is really talking about some stuff over on the right. Okay. Let's see. So the results database was launched in response to FDAA, the Food and Drug Administration Amendments Act, and it was based on statutory language and informed by Consort and other relevant reporting standards. So we were really implementing what Congress told us to implement, and it requires that the sponsors or investigators of trials report what we call the minimum data set. And it's the data set that was stated in the trial protocol. And for our purposes, it's the trial protocol as reflected in the registry record. So you register with some summary data about your protocol, and then we say, okay, you said you were going to measure, give us the mean blood pressure in each arm, give us the mean blood pressure in each arm. It's a tabular format with minimal narrative. And also I should note that EMA, and you'll hear from someone from EMA, I think tomorrow, is developing a results database really based on our model. We're trying to be as harmonious as possible for all obvious reasons. So this is what the tables look like on the left, and basically the four scientific modules are participant flow, like a consort diagram, baseline characteristics, your pre-specified outcome measures, and adverse events. All summary data. I haven't said that often enough yet. Okay, we have review criteria. So people enter the data, and we have review criteria. We don't know what's true. We don't know how many people got better in your study. We don't actually even know if you did a study. We have mechanisms in place to hope that we know that you did a study, but we don't know how many people got better. But we we do require complete and meaningful entries, so you couldn't enter, a value of 5.4 on the Zarin scale and expect that to stand because no one knows what the Zarin scale is. You couldn't, we have checks for logic and internal consistencies. For example, the number of participants must be consistent across modules, which doesn't mean that you have to do an intention to treat analysis. We're not the trial police. We don't say what you have to do. We have to say that what you're reporting makes sense. And I'll show you some examples where 300 patients or participants showed up. So the participant flow had 400 people, and suddenly you're giving us results for 700, and it's like, great, but who are these people? Time to event must be measured in a unit of time. Things like that. Picky little things like that. 624 years cannot be the mean age. Now, we don't know if 62.4 years was the mean age, but we know this can't be. So that's the kind of review we do. So what were the initial assumptions? We assumed that the required data, so the data that is now basically legally required, were generated routinely after each clinical trial, because again, this is what the protocol says you're going to do. So the requirements are based on the protocol. We assumed the data would be necessary to understand the results of any trial, and that therefore the data would be necessary to also write a journal article. How can you write a journal article without knowing the basic things about your trial? So we thought the burden of reporting would be mainly due to the data entry and the time frame requirements, because there's sort of rigid time frames of when you have to do this. Well, we were wrong. So it turns out what we've learned is that protocols may be vague, may not be followed, and what I didn't write here is, from what we can tell, may not exist. Summary data are not always readily available, even for trials that had already been published. So, we'll have trialists will come and say, well, I published this 2 years ago, what are you hassling me for? And they'll say they don't have the data for their primary outcome measure. Well, we didn't get that analysis done. For many trials, nobody was available who could explain the structure or analysis. And what we learned is there's not an objective, easy-to-describe route from the initial participant-level data to the summary data. Many people and many judgments are involved. So again, going over our minimum requirements, and we still wonder which of these are not used for writing a journal article: the number who started and completed each arm, the age and gender of participants, which are the most elemental baseline characteristics we could come up with, each pre-specified outcome measure, pre-specified by you, the trialist, and adverse events that were collected. If you only collected some kinds, that's all you had to report. We didn't want you to invent adverse events, right? But what we learned is, and this was the most surprising to me, structural changes to study. Arms come and go. People would describe it as a 2-arm study, and suddenly they're reporting a 4-arm study, or more likely, a 4-arm study, and now they're reporting 2 arms of the study. Or arms come flying in from another study. Like, oh, we always meant to include 100 people from this other study. It's like, great, tell us who they were. Participant flow and baseline characteristic tables describe different population than the outcomes table. I never understood that. They'll say, oh, those were different people. It's like, well, why were you telling me their age and gender? I don't really care. I care about the people who are here. Data providers can't explain the denominators. In other words, they'll give us an outcome measure, and I'll say, who were these 59 people? I didn't mean their names. I meant, like, which arms did they come from? And they don't know. So here's an example. This was my sort of exhibit A that many of you have seen me talk about before. This showed up in a New England Journal article. It's an NIAID-funded study from a few years ago, and it was a 2-arm study, so I do what I do, and I looked up the clinicaltrials.gov entry, and it was registered as a 4-arm study. And then if you read the New England Journal article really carefully, you found out that it in fact said that they were only reporting the people with high viral load, and a DSMB had told them to collapse 2 of the arms. So the New England Journal article had some integrity, but for the purpose of clinicaltrials.gov, you still needed to tell us what happened in all 4 arms, right? So even though the journal article was only talking about half the subjects. It took literally, I think, 9 to 12 months of working closely with the statisticians at the Harvard School of Public Health who are under contract with NIAID to do their analysis to figure out who the people were and where the arms went, where the people went. So literally saying, how many people did you start with and where'd they go, and who is the denominator for each of these outcome measures, And these are brilliant statisticians. They were in charge of the data. And this trial was basically too complicated for them to figure out. So they were giving outcome measures without actually knowing what the denominators were. That's one kind of problem we've seen. We get incoherent entries like, 823.32 mean hours per day of sleep. By the way, I love the number of significant digits. So a lot of precision in the numbers, but like things that make no sense. Time to survive. And these are things that I've had discussions with the principal investigators about. This is not, we had initial problems where people would assign data entry to a clerk or something. But these are times when they bubble up to me. The chief investigators and the statisticians tend to be on the phone. And these are things, time to survival, where the person on the other end of the phone did not understand why that didn't make sense. 36 eyeballs in a study of 14 people. When I said, where'd the extra eyeballs come from? They just sort of said, oh, well, we didn't really count how many people were in the study. Mean time to seizure equals 18 people. So a mismatch between the outcome measure and the... These all give us, they aren't reassuring. As a consumer of the medical literature, these aren't reassuring things because, for obvious reasons. Here's an example. This was an industry trial. I have no recollection of which company it was. We were on the phone again. By the time it bubbles up to me, it's gone through many levels. And they had their statisticians from, around the world on the phone, the chief statisticians on this trial, and some investigators or someone else on the phone. And we're going over the adverse event table. And I said, what's the difference between drug and drug all? And they said, well, drug all includes more of the people. And I said, okay, why are the denominators both 297? At which point you heard the statisticians whispering to each other. And then you heard someone say, we'll get back to you. And now look, the percentage of serious adverse events in drug is 51%, and in drug all is 64%. Now you can imagine a situation like a comparator where that's an important difference, which makes you feel like anything could be anything. I actually was going to be brave and have musical snippets in this talk. First was there's something happening here, what it is ain't exactly clear. The one for this was, every hand's a winner and every hand's a loser. This made you feel like any proportion could have been presented, and there was sort of no sense of objectivity here. So examples of changed outcome measures, quality of life scales replaced by a depression scale, 1 month data replaced by 3 month data. Number of people with an event is replaced by time to event, and that's really common. We see a lot of movement between time to event data and event count data. All-cause mortality is replaced by time to relapse. These are just examples. A lot of this is reviewed in this article in the New England Journal, but now I'm going to show you the next thing we've done, which is, so all of that work and our experience with quality assurance and the results database is not, like I said, is not reassuring about the validity and reproducibility of the summary statistics. It's very worrisome because you feel like before they had to report to clinicaltrials.gov, they did not have a piece of paper with these obvious stats. And again, this is when their pre-specified outcome measure was mean diastolic blood pressure. And then when we say, tell us the mean diastolic blood pressure, they can't. And that's like, well, what was happening before you had to report to clinicaltrials.gov? So in that context, we worked with a group at Oregon, Mark Helfand, Dan Hartung, and colleagues, on a study comparing when there was a journal article about the same trial and when there was a clinicaltrials.gov entry. And we compared the summary stats, the same summary stats, and not surprisingly, 82% had at least one important discrepancy. And that's not surprising because you feel like there's a sausage factory going on, and the sausage might taste a little different each time you make it. Here's an example, a particularly egregious one, but it was maximal walking distance at 12 weeks under 2 treatment conditions, and in our system it said 618 meters, and in the publication 775, with the same denominator. I mean, as far as we could tell, it was supposed to be the same information. All I can say is they both can't be correct. Whether either of them is correct, who knows? So we have, this is along the horizontal axis is the list I showed from the beginning of types of trial data going from uncoded all the way to the analyzed summary data. And the vertical axis, in my mind, is level of information. So you're losing information at each step of the way. But also each vertical drop involves subjective and not transparent judgments. We don't know, especially if when we're talking about sharing data, a lot of times people are talking about the analyzable data set. All those judgments have already occurred, and it's very hard to know what they were. And because it's not a straight line, it's hard to imagine it's reproducible, and it's hard to know what to make of the summary data in the literature. So that's our concern. It's a big problem because what you get out of journal articles are a bunch of numerators and denominators. And it's sort of like, what if they're not correct? And if you're doing a formal decision analysis, those are the things that give your point estimates for the probabilities. If you're doing an informal, those are still the things that guide your clinical decision making. So again, the process is not transparent. And just say, this has been mentioned, Dr. Drazen's editorial about that study that I think Liz Loader showed. My concern with this is it's not what's in the data. It's not what you can read in the journal article that worries people. I think it's what you can't see. It's all those vertical drops. And providing more transparency, I believe, might help that. These are the documents that may help to explain the journey. The clinical study report is in there and includes some of the documents from above it. They may help to explain how things were done and may therefore help. So in sum, the journey from initially collected data to summary data is not completely objective. Greater transparency, I believe, could help to inspire trust. And greater transparency could also help the field, meaning all of us who are engaged in clinical research, to engage in internal quality improvements. We have found practices that no one thinks are correct. It's not what's described in textbooks, but previously no one knew what anyone else was doing in their trials. So transparency is what I would encourage. And I'll stop there. Thanks.
Let's hold our questions until the panel discussion in the interest of time and also to discuss it all together. So let me now introduce Charles Hugh Jones, who is the Vice President of Medical Affairs North America at Sanofi, and he's going to speak about the Data Sphere project.
Thank you very much, Jeffrey. Thank you very much for the invitation to be here today. So as I said, I work at Sanofi. That's my day job. For my evening job, I work for an organization called the CEO Roundtable on Cancer as part of their Life Sciences Consortium. And I just want to spend 10 minutes now speaking to you about Project DataSphere, which you'll see is an initiative to, I hope, quite boldly make data sharing happening, both at the academic and the commercial side within cancer research. And it's cancer research specifically because there are some very good reasons why we should be doing it there. So let me just start for a second by telling you about the CEO Roundtable, because it's an important organization. It's important in helping make data sharing happening. It's a group of CEOs of the largest companies in the world and some of the smallest companies as well, who look for ways of reducing cancer in the workplace, stopping smoking, health, exercise, and so on. But at the same time, we take groups of people within those organizations, the heads of R&D, medical affairs, and so on, as a life sciences consortium to work on specific projects. And you'll see here there's Covance, there's Duke, there's GlaxoSmithKline, Sanofi, and so on. And it's done under the safe harbor of the Institute of Medicine, the NCI, and the FDA. And what's critical is those CEOs are able to help us make the decisions that sometimes don't happen in organizations about can we safely let data go. So I want to talk about three things. We've got 10 minutes. The problem of oncology, data sharing, and Project DataSphere. I've got about 9 minutes left, so I feel a little bit like President Obama last night, who took 5 seconds and turned it into a whole policy debate. I don't know if you saw that. So we'll rush through this. I'm only going to give you one slide on the problem with oncology. Our colleagues in cardiovascular medicine have done an extraordinary job over the last 30 years. And you see here the mortality rate per 100,000 population for patients with cardiovascular disease has more than halved. Everything from beta blockers, ACE inhibitors, and so on. If you look at the same data for cancer over the last 30 years, you see it's a radically different picture. And you'll see that there is a slight delta around 1991. It starts to track downhill, but the vast majority of that is from changes in smoking habit and changes in screening like colonoscopy, PSA, and so on. And only a relatively small part of that is due to therapeutics. That's not to say there hasn't been huge and very effective progress in the cancer marketplace, but we're just not being as effective as we would like to be. And at the same time, the cost of cancer drugs over the last 30 years has skyrocketed. And we all know the problems we've got with healthcare. We talked about this in the debate last night. This is an unsustainable situation where we're paying in excess of often $100,000 for a cancer treatment. We need to find better ways of doing it. One of the solutions of many that we need to address is data sharing. So this project is specifically about how do we make data sharing happen within oncology. I think I'm preaching to the choir. Everyone here obviously believes that data sharing is something that's particularly important. We wouldn't be here otherwise. I think the single biggest driver should be the fact that 7.6 million people die every year with cancer. And there's someone, or every one of us knows someone who's been touched by cancer. But then there are the more prosaic reasons, faster, more efficient research. Certainly in cancer, we're getting much better at subtyping individuals who are getting different types of cancer and turning what were formerly large cancer groupings into much smaller disease areas. One of the problems you have with that is you start getting fewer and fewer patients, and that's important from a statistical methodology point of view. And then there are issues related to transparency, real-world data, unknowns, and so on. So everyone's on board. The JCO says we should be doing it. Peggy Hamburg at the FDA says we're doing it. There's vast quantities of unused data, and yet no one is sharing that data. So what is Project Datasphere? Well, the fact is the need exists, so why won't they do it? And this is what we looked at, first of all. What are the problems to making data share happen? And there are obviously unique challenges in healthcare, privacy, and so on. But the attitude seems to be, and I'm quoting Andrew here, that don't share unless I can prove that no harm occurs, and we need to turn that round. There are academic disincentives. The whole tenure system within universities is based around not sharing your data, keeping it as close to your chest as possible so you can publish. From a patient point of view, you're concerned about privacy and ethics. At a corporate level, IP. And then everyone actually has problems with what are the resources needed to actually do the data sharing. It does take time. So we then said, well, what would great look like? And in my idea, a great data sharing would be a library where you could go into a room on lots of shelves, as it were, metaphorically. This is electronic. But you could go to different shelves and say, I want to take down the books on this type of breast cancer, or maybe for a particular pathway, let's say PARP inhibitors. And you can take the data set, plus the blank CRF, so you know how the data was collected, plus any other documents like the protocol, and take them into a working room in the library and then start to merge the data around. That would be my ideal situation. So we tried to work on this idea and see how we could make it happen. So the first thing, it needs to be simple. It needs to be the iTunes, it needs to be the iStore of how do you do data sharing. It's far too complicated at the moment, and I think that creates a barrier. You need to have systematic sharing. And for us, the single biggest goal at the beginning is getting access in the data. I'm 100% on board with data standards. I'm working very closely with Becky Cush at CDISC. But first of all, we need to get the data out there. So what we're proposing here is initially we're going to put comparator arms, and I put a caveat there because I'm really working towards getting active arms and genomic data, the protocols, the blank CRFs, and the data descriptors so you know how this data was collected. Collecting data from industry and academia, positive and negative studies, they're all good. It's all good data. A negative study often tells us much from an epidemiological point of view as a positive study. And put it into a publicly accessible, and again, publicly we can debate here. My principle is as open as possible, in a simple web library that respects the privacy issues. And then use the convening power of CEOs together with patients and advocacy groups. And this is where communication is going to be one of the single biggest things we have to do to make this happen, to drive academia and industry to share their data. And then share it in a standardized de-identification way. So when people come to me and say, how do I do it? I can give it to them in a bundle and say, this is how you de-identify your data. If you do it this way, you'll be fine. And then work with third-party data aggregators who can then take the data that we've got. They can take books off the shelves, take them to the tables in the library and start working together. I'm not going to have any judgment call. We as a team are not going to make any judgment call about how you should use this data, what you're going to do with it. Our job is to provide the data. And the reason for that is critical within oncology is there have been some very successful applications of aggregating data. For example, in Alzheimer's disease and Parkinson's disease, the problem we have with oncology is there's something like 800 drugs being developed, about 9,000 studies in clinicaltrials.gov. The reality of us being able to aggregate all those data sets ourselves is impossible. So what we need to do is use the crowd, use the crowdsourcing approach to aggregating this data. And so that's how we came up with what is the data sphere. So we need to make it easy to do. So we asked 3 questions. Why would people even want to share their data? The second question is, how would I do it? So if I was prepared to share my data, how would I do it? And once I've prepared my data and made it relatively easy to post, where would I put it? So we've set about creating or defining the incentives for donors related to productivity, and we've built a model in terms of potential cost savings, demonstrating that you can get increased citation from sharing your data, and then potentially actually getting a DOI number attached to your data, so FDA can then use it as part of their regulatory aspects. Persuade donors that there's a real benefit to collaboration, and then start to look at ways of micro-attribution. And we're taking some software that's been used in the insurance industry for doing attribution between different insurance companies and seeing if we can turn that towards attributing folks in the science realm. At the same time, we have to incentivize patients, and that's again all about communications, how to define that we're using their data and we're using it safely. And then I don't think we have to incentivize the researchers as much. If we provide them the data, I'm sure they'll get on with it. But I've had some great discussions with companies like Kaggle, where you can actually run data competitions. I love their strapline. It's making data science a sport. Then there's the how. So it's coming up with tools. We're creating standard de-identification, working with Vanderbilt University, a single data use agreement that everyone agrees to. And you can fill that in online. And then the resources in terms of how-to guides, a sort of service guide for doing your de-identification, forums and so on, and then tools for advocacy. And then lastly, we're working very closely with the database company SAS, and they've been incredibly helpful doing this work pro bono for the CEO Roundtable on Cancer to produce a secure, simple, powerful, scalable website where fundamentally everything you need to do to share data is automated. Everything from signing up to access the data to downloading it or uploading your data use agreements, and so on. So I'm going to finish off by showing you this is the first iteration of the database. I spent a couple of hours on the phone with SAS yesterday looking at the incredible work they've done generating the database. All sorts of tools tucked away in About Us, contributing data, guides for use, and then really educational tools for the public and companies about sharing data. So here you'll see some of the aspects. And then finally, what we're going to do from Sanofi is we're going to donate 2 recent phase 3 clinical studies, one for a drug called cabazitaxel, which was approved a couple of years ago, and then a second data for a failed drug called aniparib, which was allegedly a PARP inhibitor for triple-negative breast cancer. We've de-identified those data sets, and we're using those as primer data sets to go in the database. And the goal is to have at least 30 high-quality, and these are really high-quality company-collected, CRO-collected data sets in the database by the end of the year, by the end of 2013, and then expand beyond that. So just one quick word on our de-identification. We've used a modified safe harbor de-identification approach with Vanderbilt University. We're slightly less restrictive in some areas, slightly more restrictive in other areas, but the general approach demonstrates that we've got 0.00029% of the US population is unique. That makes us 100 times safer than the safe harbor approach, and I think that's a reasonably comfortable approach for going forward for the type of de-identification, while still keeping it useful. We're keeping dates in for the oncology research, which is obviously critical, but at the same time we're making it sufficiently de-identified in terms of HIPAA. So we're working with a huge number of partners, and I really want to thank everyone that's up on this slide. Advocacy and the social media is going to be a critical part to driving this. I truly believe this has to be driven by patients in collaboration with academia and companies to do it together. And I think with the sort of environment we've demonstrated here, it's something that can be successful. So I'm going to stop there. I this is the plan for the next 5 years, which you can see later on. The critique is we've still got lots of stuff to do in terms of microattribution, how we handle genomic data and so on. Certainly at Sanofi Oncology, we're committed to putting all the data out on the web. We're starting with these 2 data sets. We're going to move forward rapidly with additional data sets. So finally, just to acknowledge the various members of the team who've been working on this project. It's not just Sanofi, it's Pfizer, it's Celgene, it's SAS, it's Duke, and so on. So thank you very much, and any questions, there's an email address.
Thank you very much for that visionary description of the DataSphere project. I want to move on quickly then, if we may, to the Yale Medtronic experience, and let me introduce with pleasure Richard Kunz, who's the Senior Vice President and Chief Scientific, Clinical, and Regulatory Officer at Medtronic.
Thank you. Good morning. What I want to do is share our journey for transparency. It's essentially event-driven, and I'll give you a brief history here. We have a product called Infuse, which is bone morphogenic protein used in spinal fusion. It's indicated in 2002 for single-level anterior lumbar interbody fusion. And what that means is that when you have a disc problem in your lower back and you've made a decision to have that removed, often the treatment of choice is to fuse the 2 bodies together. And bone morphogenic protein is an alternative to harvested bone graft. This was a successful product over about 10 years, and the base of evidence for the FDA approval in 2002 was 1 pilot randomized controlled study and 2 pivotal randomized controlled studies. Now, there were peer-reviewed publications of those articles, of those the data sets afterwards, after 2002, including additional studies done. And they included 3 more pilot RCTs and 4 further pivotal randomized controlled studies published after the approval by the Food and Drug Administration and beyond the evidence base in the peer-reviewed literature. All of these studies were sponsored and supported and funded by Medtronic. There were also studies done on non-ALIF indications, that is, off-label indications under IDEs, for potential expansion of label, and they include 2 pilot RCTs and 3 more pivotal randomized controlled studies, also published in the peer-reviewed literature. In June of 2011, probably 15 months ago, there's a major challenge that was raised regarding the validity of all of the published literature on Infuse. And the principal focus of this challenge was the results presented in the peer-reviewed literature compared to the FDA data on file in 2002. And on the general study designs and endpoint concerns and so on. So this was interesting because when we talk about peer-reviewed processes being the solution for what happens when data is transparent, the basis of our journey was that peer-reviewed journals were wrong. So this was the challenge that was made. We quickly looked at this challenge and reviewed our data to make sure that the dossiers that we had, we could verify were true. After all, this is almost 10 years ago. And we're convinced that the data was good, and talked to the FDA immediately to make sure that they felt the same. And the remaining challenge was focused mainly on how was the information disseminated in the peer-reviewed literature, what was our role as funders for this therapy, and the fact that many of the authors on the papers received large royalties from Medtronic for other products that they had helped derive. So you can imagine it's kind of a large conflict of interest matrix here. So within a few days, because there was a large media issue raised, and we were on the phone with a lot of major media outlets immediately, and my boss, the CEO of the company, this was his 2nd week on the job, by the way. So we had to make some quick decisions very, very quickly. We contacted Yale, Harlan Krumholtz and Joe Ross, who's here, because of their interest in data transparency and their capability to be able to coordinate a process that we thought we would be able to satisfy the public trust on. And what we announced was that we would work with Yale. Yale announced its process to take our patient-level data and send it to 2 independent systematic reviewers. So we agreed to supply Yale with all de-identified, and there's a long process for this and a very expensive process, and it's not scalable, as we can see yet, de-identified data patient-level data for all of the studies I showed earlier in the slide before, including all the non-label studies. We also provided all FDA correspondence and all adverse event reports, and these are thousands of reports. So we tried to make sure that there was nothing that anybody would reasonably want to have that we wouldn't supply to Yale. We then agreed so 2 levels of transparency that we wanted to work with. Our main concern was addressing these issues, because if these issues were important and had credibility, we wanted to know them and we wanted to alert the public. The second thing is we also wanted to make sure that we had trust with the public, because we sell products that they use when they're sick. And at that time, we agreed that the data would go completely transparent to 2 systematic reviewers for them to do an independent review. And then we wanted to take one step further with collaboration with Yale to provide this into the public domain. And that process, we're in the final phases of understanding how that's going to work. In the fall and winter of 2012 is when we'll have the publications of the systematic reviews, and they'll be in 2 forms. One is a formal review, and the second will be a manuscript that will be published in each of the systematic reviewers in the Annals of Internal Medicine. And we're almost in the phases of that being submitted by the systematic reviewers. And again, we're completely hands-off in this process. And that's where we stand. The actual process for the transparency component that we've committed to and is going to be determined by Yale, not by Medtronic, is still in the final phases of understanding what that structure looks like. I'm sure a few of the Yale slides about open transparency, because these were what we aligned with in our theories about transparency. The project, specifically for this project, was an extension of the Yale Open Data Access program, which included the Yaleys on the left, Harlan, Gary, and Joe, and then a special group assembled for this Medtronic Yale project, headed by Kevin Bozick and Zeke Emanuel. The rationale we've gone over. It's important to get data out. A lot of data isn't published. We wanted to both find out what the truth was, address every one of the issues were raised in the challenge, and participate in the process of making data more publicly available. The focus on industry is important because industry still provides the vast majority of studies that are published. We support them, and there are a lot of reasons why. We've all spoke about that early in the session as to why data needs to be transparent to the public. And I'll just move on quickly for this. The goal of the project was to promote the notion of clinical trial access, but to work through the kinks. And there are a lot of kinks, believe me. We spent a year working through a lot of these issues about how to go forward. And to bring together the ecosystem of physicians and patients that can share in the data. Now, I'm not going to go through all of these details here, but I'll show you 2 work streams that both Yale had and we had just to get this project together. The Yale project work stream was mainly focused on how to take the data, assemble it, and give it in the most unbiased way to 2 systematic reviewers to allow them to analyze the data and do a full systematic review. And you can see that there are a lot of processes here about how to take the data and send it out to coordinating centers. And for reasons of time, I'm not going to go through all of these processes. They're very straightforward and they're logical. Why should industry participate in this? This is something we aligned with Yale. It allows fair and objective assessment of product research data as opposed to speculative analysis based on incomplete data. We think that the best way industry participates is not to compete on how we disseminate the data, but more about how we advance our technologies. And so a level playing field, I think, does advance those individuals or companies that actually can advance patient care and not to worry about the cover or veil of data impropriety. Yale, as part of our contract, we want to make sure that this process, if it had bumps on the road, was going to be evident to the public. So Yale put together a handful of publications. We've had a few meetings about what it means to be transparent for all of the obvious reasons, some of which are we're all concerned about misuse of data. So we want to make sure people understand there is a possibility of misuse of data. That doesn't inhibit us from getting the data out of there, but one has to start having conversation that if you start to see spectacular results from somebody who gets data, think about it for a second there, because there are a lot of issues about how data is analyzed. I want to talk a little bit about the process we had to do. This is our slides. We agreed that it was important to have transparency and independence. The communication process was very complicated to make sure that we communicated the right stuff to the Yale and also to the systematic reviewers. We did not want to review to send them previous reviews or interpretations of the data, including systematic reviews that had been done before by Blue Cross and Blue Shield and others, because we didn't want to bias or taint the information. The query management process about how the data would work between systematic reviewers if they didn't understand a piece of data they got, or they wanted to validate that actually the data was real and come. There were processes we had to have to make sure that they could be handled in an unbiased way that could be audited and transparent. That had never been decided. There were no precedents for that before, so we had to invent these. We don't know if they're the right ones. We think they're pretty good. The de-identification process, we talked about that earlier, is complicated. There are 18 HIPAA fields that have to be de-identified. We could de-identify about 15 of those fields. We had to hire an expert in de-identification. I'll go quickly and I can show you that the processes were expensive and long. This took about 4 months to do with about 25 people to de-identify these data sets. The total cost of this project is about $2.5 million. The process also, we had to de-identify approximately 1,500 medical device reports from the Food and Drug Administration as well, and they had to be reviewed. We had to hire an expert, statistical expert in de-identification and make sure that they were certified, and that had to be addressed with HIPAA lawyers and so on. So it was a big process. I want to get this slide very quickly. A couple of concerns that we still have, and they're not major concerns. They're, I think, issues we need to raise. And this is based on the experience we've had in trying to do this process for over 14 months. If we make a transparent process for the data and make it available to the public, the query aspect is, do we want to know who's asking the question and why? And is there an interest in the truth? A lot of concerns about what if a litigant firm wants to get the data and they identify themselves as a litigant firm. Well, litigant firms can also find the truth sometimes. And so we know that they're incented to find things that are going to raise money. But at the same time, what's the concern about that? And we don't want to limit somebody who wants to find the truth. But we know that there are a lot of alternative reasons to analyze data in other than truthful approaches. What's the question? Can we have an independent group that says, is the question worthy? Does it serve the public or not? Or is this a data dredging? And what methods are you being used? And should they be pre-specified? If you're going to ask a question, should you say, how are you going to answer them? And that should be reviewed by an independent panel. Access. Should there be an initial zone of propriety and time? That is, that should we or academia or anybody who's going to share data have at least a year so they can get their stuff done before it's sent out? That's not really well worked out. Anyway, I think we touched on that this morning. What level and portion of data is requested? Is it all of the data? How deep do we go? Do we go into the adverse events and so on? These are complicated. Should there be a time limit or license on data access? If we hand you the disk, are you going to keep it forever and do a lot of analyses, or are we going to license you with a question? This is what they do in the UK, by the way. They get a single question and a time period to analyze that if you get NHS data. So these aren't worked out yet. Who controls the data distribution? The methods. Are there a priori questions on hypothesis to be tested? Should we ask people, what is there a priori question? Is there an interest in data exploration? Maybe you're saying, I actually want to do a data dredging experiment. Well, should we let them do that? How do we control multiplicity and type 1 error? The analysis. Is the requester competent to do analysis? This is something that's evaluated also by the UK. They determine whether or not someone has a track record of doing analysis. If they don't, and the question is merit, they will send it to somebody else, and I think they contract with Bristol to do statistical analysis. And this has to be worked out. Should the analytical methods be transparent to the public? Should you list your methods and then should be tested to make sure they're there? What about secondary data sharing? Can the requester share the data? That's an issue that hasn't been worked out either. And believe me, that if someone gets a disk, it will be on the internet. There's no question about that. And dissemination of the results. Should there be controls on the results dissemination from the secondary or primary analysis? Do we allow unfettered dissemination of the data? Or is the dissemination only after a peer-reviewed publication? That is, you can disseminate your results if you get published in a peer-reviewed journal, which isn't perfect, as we've seen, because the basis of this is peer-reviewed errors. And finally, what's the full methodological review required? Should you have a dispassionate person to review those reviews and so on? So these are the things that have bubbled up in our feeling because we're in the middle of this for the last year or so that have driven it. And then my final slide is one thing I also want to address. It's important, and I hope we touch on this, is we're now being asked and to understand what is our role as industry compared to the peer review role, okay? I won't read all the things on the slide, but and the bottom line is we have a regulatory responsibility to do studies with the regulatory agency to produce those results in a faithful and trusted way and to disseminate them under the law. But we work with academic investigators who also publish in a sphere of peer review. And that sphere of peer review doesn't want to have industry involvement at all. So if we have investigators we work with who decide to cherry pick endpoints that they want to put into tables, to potentially modify the protocol and analyze what they've seen and some of the stuff that Deb talked about, what's our role to police that? Should we police that or not? Or is that a different, or should the peer review take care of that per se? These aren't worked out at all. And I think we'll probably address some of these issues if, in fact, some of the challenges about peer review problems or misuse of data that was challenged by the principal investigator about how it was disseminated, then we will come back and say, this is a product we actually sell to people. We're responsible for the health of those individuals. What is our role in policing the academia peer review in that situation? And that's not what worked out. And I'll stop there. Thanks.
Thank you very much for presenting that very challenging and visionary approach to data transparency. We're going to shift gears a little bit now and talk about opportunities and models that foster pooling and analysis of data. The first presenter is John Wagner from Merck, who's the Vice President and Head of Project Management and Early Development Projects.
Thanks very much, and to the organizers for the kind invitation to come here. The disclaimer is these are personal impressions. I'm not representing the Biomarkers Consortium nor Merck, but most of what I'm talking about is actually published. Just a word about pre-competitive collaboration. The pre-competitive collaboration is something that the biomedical enterprise freely stole from the software industry. And why? Because it's something that probably works pretty well in increasing the efficiency of work. There's a great definition in this book by Steven Weber: competitors share early stages of research that benefit all. And I think there's a growing awareness, acknowledgement in the biomedical enterprise, in academics, government, and industry that pre-competitive collaboration works and can be used to really drive some work in our increasingly complex world. So the Biomarkers Consortium is a pre-competitive collaboration, and the nice thing about the consortium is that it's a group of institutions, companies, government agencies that have come together with a common goal for biomarkers. And there's a really key role in having our regulatory colleagues at the table, especially for biomarkers. Biomarkers are going to be used more broadly in various, possibly regulatory sorts of situations. So the goals of the Biomarkers Consortium were laid out as facilitating the development of new biomarkers, qualifying those biomarkers for use, generating information that's useful, possibly to inform regulatory decision making, and then importantly making all of this completely public. I want to talk just briefly about one biomarker, adiponectin. And this is a protein biomarker that was discovered in the 1990s, and it's associated with there's decreases in adiponectin in obesity, decreases in insulin sensitivity, and drugs like pioglitazone, PPAR agonists increase adiponectin. Adiponectin is thought to possibly be part of the mechanism for insulin resistance, and also part of the mechanism by which PPAR drugs like pioglitazone exert their effects. it's very clear that these medications drive up adiponectin concentrations very quickly in patients, in healthy volunteers, and in patients. Now, the issue with adiponectin when this project was conceived is that there was quite a bit of research on adiponectin, these associations with insulin resistance. But one question that wasn't addressed is whether adiponectin, a response in this biomarker adiponectin, could predict a hemoglobin A1C decrease in patients with type 2 diabetes treated with drugs like pioglitazone. Why is that important? Well, if there were a good predictive biomarker like that, it could be used for patient segmentation, and the patients who were not going to respond to these agents could get other therapies, and it would only be the patients who were likely to respond that could get these medications. Now, it's interesting that this was a very active area of research, and there were many file drawers and computer hard drives full of adiponectin data in various companies and in academic institutions. But the questions, the data that addressed this question were relatively sparse data that were publicly available. And clinicaltrials.gov doesn't answer this because most of these data were older and also were exploratory endpoints, at least the adiponectin part, so not really readily available, at least in any kind of an easy way. So when the Biomarkers Consortium was put together, there was this was really a proof of concept project. This was thought to be something that would be relatively simple to put all this data together and answer this important research question. So a number of companies did agree, GSK, Lilly, Merck, Roche, to put together the data, actually a blinded data set for many trials in over 2,000 patients. And we had statistical analysis that was donated by NIH and by quintiles. And this project went through a series of phases. I won't describe all of it. The first two phases were really meant to assess whether we had a good database, whether it was valid for answering the questions that we really wanted to get to. And it was the last phase of the research where we were asking the question about the prognostic value of adiponectin and hemoglobin A1C. We worked through a project team and then made those results publicly available very quickly. So this is basically the result of all of that work, and it turns out that adiponectin is correlated with hemoglobin A1C lowering. That correlation isn't as good as fasting plasma glucose. That's not really an enormous surprise, but it's still a very important observation because the fasting glucose changes occur very late in treatment, and the adiponectin changes occur relatively early. So even with a modest correlation like this, there was really a hope that this could be used as a predictive biomarker. And that's shown in a receiver-operator curve kind of a model in which the receiver-operator curve characteristics of fasting plasma glucose and adiponectin are pretty similar. So we concluded that adiponectin is a robust predictor of glycemic response to these PPAR agents. We confirmed a lot of previous findings in this aggregated database and really concluded that the potential utility of adiponectin across the spectrum of glucose tolerance was well demonstrated. I think for this audience, the most important conclusion was that this project established that a cross-company collaboration was a robust and feasible method for doing this kind of research. And we kept this project team going even after the result in order to really delve into some of the lessons learned. So transparently, this project, this is the timeline for the project. And you can see that it took a long time to accomplish this project. All of that data was collected well before 2007. In fact, the kernel for this project was really conceived of several years before the institution of the Biomarkers Consortium prior to 2005. And so the time it took to really accomplish this is pretty long, and that's a real problem. You can see that there's many steps along the way. As we learned about how to collaborate better, this was one of the initial projects. It was the first published project by the Biomarkers Consortium. We were getting better at it, but it did take a long time to accomplish. So we learned many lessons in this consortium. One of the issues was that the focus, organization, and pace of the group really sunk to the lowest common denominator. Now, this is a group of busy industry scientists, some collaborators in a CRO, collaborators in the NIH, and it was not always the first priority of everyone involved in the project. You have to be honest. It was the evening job for many people, myself included. And that's really a problem. We addressed that in part by making sure that we used good project management skills, and in fact this became a best practice at the Biomarkers Consortium of having a very good project manager associated with all the projects. There also was a lack of collaboration tools initially, which the Biomarkers Consortium has also addressed. We really struggled with the data sharing principles and standards. It was difficult to negotiate the data sharing plan with even the small set of companies to a level that satisfied everybody. The definitions, the standard definitions, were not all obvious. And someone else mentioned it earlier. Some people left particular companies who actually had some of the crucial information. So there was limited institutional memory. It was incredibly important for this project to have a single knowledgeable legal liaison who would be responsible at the individual companies. In fact, the knowledgeable part was really important because a lot of legal folks in large companies, this data sharing is not their day job either, and we really had to spend a lot of time explaining what the purpose of this project was. In the end, this project came to a template for the Biomarkers Consortium for data sharing plans, which is hopefully driving faster work for future projects. There also was a lot of issues with the limitations of the existing data. This was an existing data sharing project. It wasn't conceived of, the studies that we were using weren't conceived to answer this research question, so there were real limitations. There also were limitations in the Adiponectin data itself, because different companies use different assays, and we had to do some work to assure ourselves that we could really combine that data. For projects like this, we really just need to acknowledge the limitations and move on, and have prospective follow-up when that's necessary. Now, there's also some broader issues that we ought to talk about. Some of them have already come up this morning. The clarity of the question, your research question, is really very important for defining the type of collaboration. The role of a neutral convener, in this case the Foundation for the NIH, was very important in gaining the trust of all of the folks all of the stakeholders who were involved in this. Dialoguing with the FDA is incredibly important, and in this case we had an FDA representative, and we probably didn't even push that far enough in terms of making sure that we were qualifying this biomarker in a regulatory acceptable way. There were definitely behaviors that impeded this collaboration, not the least of which was just the time that the project team members could dedicate to this effort. And then some more general issues. We really observed that most collaborations, this one included, had even itself had a siloed attribute to it, and it was really incomplete. I need to point out that not all companies that had adiponectin data shared in this effort, and not all of the adiponectin data in each company could be merged into this data set. That's an issue. And the motivations were also an issue. Now, the Institute of Medicine had been involved in, through the National Cancer Policy Forum, an effort where really work some of the issues around the clarity of the question. And this is an article by Jill Altschuler that came out of some of those efforts. And we won't spend much time here, but if you look on the left-hand side there, it's boxes that represent how open the contributions are and how open the output is. And depending on the kind of question that you're asking, the openness of the contribution and the output are going to change. Also, the type of question, whether you're developing standards and tools, aggregating data, creating new knowledge, or developing a product, is going to change the kind of collaboration that you want to set up. The Biomarkers Consortium is a consortium that's labeled number 4 in this slide. That is a restricted contribution. The parties that involved in the collaboration are named and restricted, but the output is completely open. And this is a good model for generating new knowledge. I think that the key role of the neutral convener is now readily acknowledged. The role of the FDA or other regulators is readily acknowledged. But some of the other issues here continue to be an impediment, in my opinion. We really need trust and openness when we're engaging in this kind of collaborative behavior. We can improve collaborations by really being very clear about our common goals and the unique value each party brings to bear. And we really need to strive for open inclusiveness in these appropriate collaborations. All the parties with the stakeholders with the data really need to be there. And this last point was also brought up earlier. You know, the motivations are similar and different across stakeholders. If we continue to reward academic folks with a sort of principal investigator laurels, and reward pharmaceutical companies for secrecy, then we're not going to get what we want in terms of these kinds of collaborations. So finally, I just want to acknowledge the folks who participated in this. On the left are all the folks who were involved in this Adiponectin project team that was through the Biomarkers Consortium, and on the right were folks who were involved in a pre-competitive collaboration workshop like this one that was convened by the National Cancer Policy Forum. Thanks.
Thank you very much, John. Let me introduce then another program which is somewhat similar in nature that Jonathan Rabinowitz, who's the Elie Wiesel Professor at Bar-Ilan University, concerning the New Meds collaboration on psychiatry trials.
Jeff, thank you. Thank you very much, and thank you to all of the organizers. I'm going to talk to you about our project called New Meds, Novel Methods for Development of Drugs in Depression and Schizophrenia. It's a European Union-sponsored project. It's a unique project. It's a private-public partnership. And I'm going to talk to you about our repository of data. And I'd like to just ask you for a moment, with the exception of those who are listening to this as a webinar and operating a motor vehicle, or any of those in the audience who have narcolepsy, just to close your eyes for a second or allow yourselves to imagine what you might do in some therapeutic area that's near and dear to you if you had access to almost all of the data out there in your given area. And just think about that for a second. All right. Because I'm limited on time, it had to be a short second. It couldn't be a full minute. But if you could try the exercise at home, again, not while operating a motor vehicle, it could be very useful. So I'm going to tell you about 2.5 years ago, I was approached by Satish Kapoor with some people from the Institute of Psychiatry in London, who was sort of the mastermind and main academic head of the new meds effort, and people from industry he was working with to put the project together. And they said to me, would you be willing to take on heading a group that would create repository of data on antipsychotics and antidepressant data and try to mine this data? And I accepted the challenge, and I'm going to tell you about the fruits of our labor and also some of the toil involved in creating this data sets. So these are our partner companies. It's a European Union sponsored event, study under the Innovative Medicines Initiative. So these are the list of our various partners, universities, and SMEs, which stands for Small Business Entities, I believe. Not to be confused with SMSs. Okay, this is the funding and the overall goal. NewMeds has 10 working groups that vary from things like animal models, imaging techniques, and I'm going to speak about the topic of advanced data analysis techniques because I don't know anything about those other topics. So, okay, and I'm going to tell you some of the findings we had. We put together, first of all, these are our methodological accomplishments. Now, I stole the wording from this slide from the Institute of Medicine. I was invited to speak at a CNS forum of the Institute of Medicine a year ago, August, and they had set up for this meeting these objectives. And lo and behold, we had met these objectives, and I'm very proud of it on the methodological level. So we've established a consortium that shares data, and just to use the parlance that was used earlier, this is coded patient participant level data. Is that right, Deborah? Okay. From industry and academia to examine pre-competitive questions. We've overcome challenges associated with establishing data sharing. We pooled and mined data from studies that have sufficient common experimental designs to have reasonable chance of valid conclusions. Okay, we have two data sets. This is our schizophrenia database of antipsychotic drug trials. These are the companies that contributed their data. These are the pivotal studies and other studies that they had. We focused, what I'm going to present today are the placebo-controlled studies and the lessons that we learned from those studies. We also have one NIMH-sponsored study and one EU-sponsored study. So altogether, we're talking about data on almost 26,000 patients from 64 industry-sponsored trials and then 2 additional trials. We're also in the process of setting up now our antidepressant database, and this is what we have so far with additional data on its way from Eli Lilly. So far, we've mined the schizophrenia data, and I'm going to talk to you about those lessons today. So I'm going to just jump to our major findings, just that you have a sense about how this might be useful in other areas in which you're involved in. Our first focus was the issue of placebo response, which is a problem in many different areas of medicine. We found, first of all, that studies could be shorter. Most studies were 6-8 weeks. At 4 weeks, you knew what you were going to know later on. We also found out that females showed more pronounced differentiation placebo-active treatment, yet they were underrepresented in the studies, suggesting that possibly if we had more females in the studies, we might show a larger signal. We also found that a group of patients who had later onset of disease showed more pronounced improvements irrespective of their allocation to active treatment or placebo, but it didn't affect the differentiation. However, we did identify a group of patients who were under the age of 30 but had more than 4 years of illness, so they were after that critical first onset of illness group, and they showed a heightened active and placebo difference. We also found that patients that had what's known as both positive prominent and negative symptoms showed the most placebo active treatment differentiation. And we found that we were also looking, one of the lore of trials was that people who are running trials, because of the desire to recruit or the necessity to meet certain recruitment requirements on a timely basis, were possibly bringing people into the studies who were not eligible for the studies. And we looked specifically to see if there was an overrepresentation of people who were just meeting symptom level criteria. We did not find that those people were overrepresented, which was actually a favorable finding and spoke well of these studies. We also found one of the issues in these kind of populations of patients who are on antipsychotic drugs is use of benzodiazepines. These are people who have active symptoms, and there's a need sometimes to give them benzodiazepines. The question that people had was whether maybe those are being used and showing an antipsychotic-like effect, and maybe they're used more in placebo patients. We did not find that this was the case. We also found that there was more placebo-active difference in Eastern Europe than in North America. Again, this didn't surprise people who were involved, but it did confirm something. Why that is, we don't have the reasons why, but it's an important finding that we were able to confirm. Now, we put all this data together, and then we said, how could we use this in terms of thinking about designing new trials? And we used the technique of resampling bootstrapping, in which we created 1,000 sample trials by drawing patients from the studies that we had from our repository of data. So you will see over here that the current studies, the balance of patients as they are now, which is 70% female, 30% male, 20% early episode, and 40% enriched. Enriched are patients who have both high, prominent, positive, and negative symptoms. That's the current landscape, if you were, of the studies as they are. And those studies required for 90% power, about 79 patients per arm if you were to have a two-arm study. So then we said, okay, what if we use what's called enriched? Enriched are patients who have prominent positive and negative symptoms. So we would restrict the inclusion criteria to those people. Now, we're talking about drug discovery trials, so that I'm not talking about necessarily registration trials. We want to do more efficient drug discovery trials. We want to increase the signal. We want to have shorter trials, less exposure of patients to placebo and experimental treatments that may not be helpful for them. So then we said, okay, what if we just made sure there was equal opportunity of males and females in the studies, 50%. And now you're at the third line over there. So then you were able to reduce sample sizes from 79 to 71. Now what if you put that together with enriched? So you took 50% female, and then you took only patients who had prominent positive and negative symptoms. Now you're down to 60. Then if you took patients, half the patients who are early episode, those are the patients who are under 30 but 4 years after their first illness onset, you're down to 57, 53, and so on and so forth, and down to 46 if you combine the various different elements together. Now, it's quite a savings in terms of patients' exposure, as we said, but it's also a savings in terms of money. Just to give you an idea of currently costs of conducting such trials, okay? Per patient cost for a 6-week study now of an antipsychotic drug is $70,000 to $100,000 per patient. Okay, so imagine if you can save from 79 down to 46, how much money could be saved in addition to saving people from getting exposure to placebo or active treatments that may not be good for them. So, and that's just one illustration of some of the work that we've been able to do with our data set. The other finding that we have is that you could shorten the trials from 6 to 4 weeks, which has implications in terms of exposure and also in terms of how much it costs to do the study. And the less you spend on treatments that aren't going to work, the more money you can spend on developing, new treatments and other trials. Let me talk about some of the implications of our findings on future drug development that I sort of mentioned as I went through this. Trials could be 4 weeks duration, using representative enriched populations and proof of concept trials. We also think here that there's a paradigmatic shift of data sharing as an ethical imperative. We feel that studies should be valued, should be given extra credit in ethical review if they're willing to share data. I can envision, for example, that there'd be institutional review board forums that have a box. If you check it, is the data going to be shared afterwards? If the data is going to be shared afterwards, then one could argue the study has more ethical validity than if the data is not going to be shared afterwards. What was that? Okay. Okay, let me talk a little bit about, in the 4 minutes and 21 seconds that I have left, there's a clock ticking here, but I did bring a quarter with. I can buy some more time on the meter. So that talk about some of the facilitators of our effort in getting together within a very short time data sets from fierce competitors in a very competitive business. Well, the companies had united around this external funding of the IMI, which was focusing on this innovative medicines initiative in a time when industry was having a very hard time with developing CNS new drugs, particularly schizophrenia and depression, and companies were leaving. So there was distress that created a common need. There was a recognition by industry that drug discovery was becoming more difficult. And one of the other facilitators was within the companies to have a clear message of support, because it wasn't just enough that the VPs of companies got together in London with us and they agreed to participate, that they needed to give this vision and transmit it within the company. And there's resistance within companies at various levels, and some of them might seem mundane levels of the people who need to go find the data, who can say, oh, well, I have an emergency thing, we have a study report due, we have data for the FDA. There are hundreds of reasons, if anybody's interested, I can give you a few over lunch, why not to cooperate, a low-level person, and find the data and give it. One of the other things that was helpful was previous relationships. I had worked with people in the companies before on various projects, and that goes a long way to fostering the kind of trust that one needs to give over this data, where the understanding is that we're not doing cross-product comparisons, but we're looking at ways of beating placebo. But the data could be misused, and that was something, a trust that we needed to have. Another thing is that we had active collaboration. I have a board, my study group, and we all talk together about the various questions, and they share in interpreting and understanding the data and the data results as well. We also had peer pressure among the companies. If one company gave in all their data, the others wanted to follow suit. They didn't want to feel inferior in terms of their performance in this realm, and we certainly use that to the max. There are a few challenges in the minute and a half that I have left. Locating data, it might sound mundane, but locating data sets in companies is very complex. There were companies that were products that were bought by companies and so on and so forth, and to locate who actually housed this data was really almost the work of a detective needed to do it. And there's also competing internal resources and priorities. We were not necessarily the first priority, although Perhaps because I was the biggest nuisance on the block, I did get the job done. I'm not sure. What's that? Yeah. Yes, indeed. Okay, thank you. Anyway, so these are, let me just talk a little bit about the future with the 41 seconds left, which is part of the future. What if all trials were stored in a uniform way and patient-level data routinely entered into a data bank? I mean, we have almost all the data here on second-generation antipsychotic drugs, and it's really a wonderful data set. Imagine if we could do this routinely. Who's best positioned to do this? Perhaps the regulators are, because they get the data. How does data sharing or unwillingness to share data impact the risk-benefit ratio of conducting a study? Well, I argue that if you're not willing to share your data, your study is inferior ethically than if you are willing to share your data. And finally, I'm at -2 now. Our experience is that the common good can be greatly enhanced by sharing the data. Thank you very much.
Thank you very much for your excellent work and your visionary talk and your attentiveness to time. The present and the future. We're going to shift gears now to have a panel discussion, and let me just introduce everyone one at a time. Sort of short presentations with no slides, and I just want to check how John Ioannidis is presenting. He's online? Okay, welcome. So our three presenters are Jennifer Geeter, who's a partner at McDermott, Will, and Emery. John Ioannidis, pardon me if I mispronounced that, who's a professor at Stanford, and Kelly Edwards, who's associate dean and a professor at University of Washington.
Well, good afternoon, everyone. I feel a little bit like I think it's my son's Sesame Street of which one of us is not like the other as I look at the amazing scientific credentials of folks who are here. And I may be the lone private bar attorney invited to join. And so I really appreciate the opportunity to come and speak with you. I think I have a slightly unique role. I was asked to have a slightly unique role in the day's presentation to talk generally about some of the challenges that we're seeing from a wide range of stakeholders: academic medical centers, community hospitals, pharmaceutical companies, biotech, and some of the legal and regulatory challenges and opportunities that surround the work that you're trying to do, and to sort of touch on a few items very, very briefly. And I'm happy to explore these with you in greater detail, during lunch. The first is that I think we have some competing public priorities. The stakeholder that one of the stakeholders that I haven't heard about so far has been the public and their demand for biomedical innovation. They go into the doctor's office, they want a new treatment, especially for conditions where the treatments are lagging behind or we're having difficulty cracking that nut. The public can be very powerful in pushing for data sharing and for biomedical innovation, but at the same time, the public remains deeply afraid about privacy. It's hard to understand exactly what they are afraid about. Are they afraid that their information will be used in ways that they don't intend? Are they afraid that their information will be used in ways that have adverse employment or insurance consequences? It's difficult to know. But everyone out there perceives a real privacy concern. And so you have a disagreement within the public about what our default settings, so to speak, should be with respect to data sharing. And one default setting would be a presumption of non-access. where in general the data stays at rest, it's only used for a limited purpose, and you'd have to overcome a presumption to have access to that data. The best example would be your classic doctor-patient relationship. This protects privacy. It errs on the side of protecting privacy, but obviously is a significant impediment to data sharing. The second model would be sort of a rules of the road model. You expect the cars on the highway to be driving, but there's an agreement about whether the on-ramps are on the left side or the right side and what side you drive on and what speed you can go. You start with a presumption that the data can move, and you focus your energies not on stopping the data, putting it at rest, but creating some safe driving rules. And in listening to my clients and coming to conferences and teaching at Georgetown, I hear both. I see both in the newspaper on the same page from sometimes the same constituents. So I think partly what you're trying to do is you're trying to do something in an environment in which there's some public ambivalence. And if there are ways to resolve that public ambivalence, then I think you'll have a real cheerleader for some of the data sharing that you're trying to do. You also have a legal uncertainty challenge. So first of all, there are active rulemakings going on right now that specifically touch data sharing. The first is active rulemaking for a final rule under the HIPAA standards. And the HIPAA standards really, it's okay, thank you. The HIPAA standards really matter for what you're trying to do because they control research activities when even it's just the data that's in play. You don't have to have a real-life person in front of you. You don't even have to be gathering new information. If you're looking at existing data and that data is above a certain level of identifiability, which is actually fairly low, then HIPAA is going to be in play. And why I say that identifiability standard is pretty low is that dates of service alone, without any other information, render that information identifiable. So if I say that a patient was treated for schizophrenia on October 1st, 2012, and I tell you nothing else, and I am speaking as a covered entity, I'm a healthcare provider, I have shared protected health information, and I can only do that in a way that HIPAA permits. And HIPAA does have specific data provisions. We expect a final rule to come out any day. The proposed rule came out in July of 2010. So we have been waiting for that rule every day for several years. I've given up predicting because I've been wrong every time. But eventually it will come out. It's statutorily required. And in the proposed rule, which really wasn't focused on data sharing, buried in the middle were some really important changes with respect to data sharing that would conform HIPAA to the rules that perhaps the pharmaceutical industry is more familiar with, general research principles. The second major form of uncertainty, and I just really want to stress this, is rulemaking right now to change the common rule. Just by a show of hands, how many folks in here know what I mean when I say the common rule? Okay, so that's great. So sometimes when I speak to pharmaceutical and device manufacturers, they're not as familiar with it because they don't do federally sponsored research, so they haven't had to live within that environment. That rule is getting a massive upgrade for the 21st century, and we're only at the advanced notice stage, so we have 2 more cycles to go. But there were some really important changes, proposed changes, with respect to rulemaking, and one was whether or not biospecimens, human biospecimens, could ever be considered de-identified. Right now, if you strip away enough information and all you have is a piece of tissue, it's not a human subject anymore. If you say that that information can never be de-identified because it contains genetic material, you lose that safe harbor of sorts. And obviously there's a huge debate within the scientific community about whether that's forward-looking and that eventually there will be enough cross-referenceable genetic information that that's a sensible standard. And then obviously folks on the other side say, if you take away the ability to de-identify biospecimens, how are we supposed to do future research with it? How are we supposed to share information? Another important I'm not sure how to, oh, there we go. Another important regulatory thing is we have a lack of harmonization. On data sharing specifically, the FDA, the Common Rule, and HIPAA do not agree. They approach this topic differently. The FDA, for example, is used to doing trials where you have a real live person in front of you. They're less accustomed to trials in which you are using leftover biospecimens. They do not have in their regulations, for example, a carve-out that if the tissue is de-identified, it's no longer a human subject, or, for example, the ability to get an IRB waiver. So far, the FDA has dealt with this issue with enforcement discretion, meaning we agree that what you're doing is a violation of our regs, but we're not going to enforce those regulations under certain limited circumstances. But it means when you are doing data sharing trial preparation, in the United States at least, you could be referencing at least 3 federal rules and perhaps state-by-state rules. And those state-by-state rules tend to be disease-specific. The analogy I always give is it's like when you cut a tree and you can see the drought years and the really wet years based on the size of the rings. It's a little bit similar with state law. You can see when we were concerned about HIV regulations come onto the state radar. We can see when we're concerned about mental health, those regulations come on, and they differ by state. And the last legal uncertainty point I just want to mention is the ongoing uncertainty about Part 11, which is electronic data vis-a-vis FDA submissions. As I'm sure all of you know, certain parts of Rule 11 are currently enforced, certain rules aren't. The reason I think this relates to data sharing is principally when you guys are doing data sharing, you're doing it with electronic data and want to be able to use that electronic data in your future submissions. I feel like I'm an auctioneer here, talking as fast as I can. You guys are gonna have to slow down the tape. It's gonna be like the old chip mix when you listen to the audio recording. On the biomedical innovation, a couple of quick things. The prep to research exception, by show of hands, how many people know what that is? Okay, so fewer. Hey, the prep to research exception is a really important HIPAA pathway that allows you to mine data in order to prepare trials. So we're talking a lot about data sharing for actually running your trials. But if you want to mine that data to say, hey, are our different data sets compatible? Do we think we could do the types of collaborations you're discussing? Do we think we have enough patients at a particular center to enroll them on a trial? This is typically the pathway you want to use if you are a covered entity. But here's the hook. To use the preparatory to research pathway, the data cannot leave the premises of the covered entity. This made sense when you were looking at dusty paper medical records. It doesn't make as much sense, in my view, when you're talking about electronic data that may never be at the covered entity's premises to begin with, because it may be maintained by a cloud computing EHR vendor or a remote EHR service provider, by a business associate, by any number of assistant players that should be housing those EHRs. Another important thing are the CLIA lab rules. They are going to regulate laboratory developed tests, and they also are not harmonized. And one of the rules of CLIA is that if you are conducting research on a new laboratory developed test, you cannot share the results with the patients enrolled or their physicians until you validate the test and then rerun the test. So imagine being a physician who says to a family, we want to enroll your child in a clinical trial. It's a very rare condition. We may see a signal of what's going on that could affect your decision to have future children, how you might treat this child, but we're not going to be able to tell you. I think this is not intuitive to folks who do research a lot of time, who may suspend sharing results for a short period of time while the trial is going on, but assume eventually they can data share with the patient. CLIA takes a different approach. And then with respect to contractual constraints for multi-institutional collaborations, let me just run through a couple. One is competing ownership provisions. We have the data that comes into the trial, but we also have the thoughts and the discussions and the ideas and what other trial we could do and how you write those provisions. There's often a real disconnect between the scientists and the tech transfer and the IP groups within the hospital. So when we talk about stakeholders, it's institutional stakeholders, but then it's stakeholders inside the stakeholders. And make sure that you're talking to all the ones you need. I've seen many, many times you get through to the physicians, you may even get through to a vice chair for research, and then you come to a grinding halt with the IP council, with the IRB, with other types of stakeholders. Think about how the trial will be paid for and how funds will be shared, not just to stakeholders, but stakeholders within stakeholders. When you're dealing with academic medical centers, you may be dealing with a faculty model, but as you branch out to different types of institutional settings, that won't necessarily be the case, and the doctors are expecting to be paid for the time and effort that they spend. And then finally, I would say, really think about the scope. What question are you asking? How broad do you want this collaboration to be? Is it every trial about diabetes? Is it certain trials about diabetes? Are you going to have a scientific board? And how are you going to avoid cherry picking? They don't want to bring the ones where they might have real IP, fruitful IP down the line, and so the collaboration tends to get watered down. What type of structural protections can you build into the contract for the rainy day? I mean, one of my hardest jobs as an attorney is that when these types of collaborations are brought to me, everyone's really excited about them. And everyone has great hope for them, and no one wants to talk about what happens if you guys stop playing well in the sandbox. But oftentimes people stop playing well in the sandbox, not just toddlers. And if you can try to build in some stopgaps along the way to right your ship as you go, that would be great. Can I talk for one more minute? Okay, yes, sir. I'm going to be indulged and be allowed to talk for one more minute. Okay, the last thing I want to talk about is clinical care, because this may not be on your radar because we're thinking about research, but especially with EHRs, the line between clinical care and research continues to blend or bleed or get blurry. And the reason for that is because a lot of the data that you guys might want to mine is going to be in EHRs, especially as the EHR standards evolve. You can think of the rollout of EHRs as a phased process. We're in 2 out of 3. And the third stage has a lot of the specifications that are about interoperability, that are about data sharing, that are about one system's ability to talk to another. And that's where you can start to have some systematic data collection across institutions. And so you're going to see data collected from clinical studies, clinical care, not necessarily from past research studies, being available for mining. There are going to be certain limitations on that you're going to want to be aware of, and that's going to be situations in which we, for other competing public priorities, have placed limitations on the ability of that data to be viewed and shared. An example would be patient safety organizations where we want physicians to self-police, and we want physicians to troubleshoot the care they've provided and not make that data available and protect it from discovery. So I think you guys are asking the right questions. And I think you will see as the regulations shake out some new questions, but hopefully some resolution.
Thank you very much. Thank you very much. We're running a little bit behind. I hope that's not our next speaker giving us a busy signal.
We're going to try one more time.
We could possibly move on to the third one, if that makes sense.
Yeah, we just want to try one.
Why don't we go ahead and move on to Kelly Edwards, if that's all right, and then we won't get any further behind in time while we prepare. Thank you.
Well, thank you very much for inviting me here to contribute to the conversation. And it's a real pleasure to be with a group who's both embracing the idea that it's not a matter of whether we should share data, but how, and really digging into the problem solving around how we might make this happen, and thinking through the things what is getting in the way of our sharing. If it's such a great idea and there's all these ethical and practical reasons why we must do it, why aren't we doing it? And I think the technical and regulatory issues are certainly significant. And I want to just spend a few minutes talking about the cultural issues, because I think these also warrant some attention. And what's a second pleasure of being with this group is, while I believe I'm the only ethics person on the docket, ethics and culture have been throughout your remarks today. And so I'm just going to amplify much of what is being said on the program. So many people have pointed to this idea that trust is really at the heart of helping to make these data sharing efforts happen. And this is trust across our relationship. So think about this is researchers trusting each other, organizations learning to trust each other who haven't traditionally done so. And essentially participants trusting the researchers and trusting those organizations. So how do we get there? I think traditionally in research relationships, we've relied pretty heavily on contracts to help us manage these trust relationships. So we've generated these crazy consent forms to really outline everything so that we're on the same page with expectations around what participants are getting into with a trial. We have data use agreements and terms of use, other contracts that manage our research relationships. And I think these are certainly necessary. I would say they're necessary but not sufficient when we're thinking about ethical research management. So I really want to put on the table another way of thinking about approaching our research relationships as essentially relational. And almost the contracts come in as a way to punctuate what we've already agreed to and what we're already understanding, rather than the sum total of how we should approach each other. So the ways we've learned from both empirical study and philosophical thinking around when do you trust someone? Think about your own relationships, and when you trust someone, there's a number of different elements that might go into this. Sometimes we trust someone who we haven't even met, but we trust them because we share core values or we share commitments, and we recognize that we're working towards a common purpose, so we can trust each other, even though I don't know you very well. So sometimes on that very common basis, we can find a path towards moving forward. Another way we often trust each other, this is seen in many medical relationships, is I can trust you if I know you have my best interest in mind. So think about how this is true in our research relationships. How is it true that we might have each other's best interest in mind in the way we approach our efforts? Another way to establish trust can be, I don't know you, I'm not sure that your statements of values and mission are that you have enough accountability in them. So I need to look for your rules, those rules of the road that Jennifer referred to. Are they clear and transparent? The way that this system is going to function, can I see it well enough to understand how it might work? And that might be one way we can accomplish trust. And then finally, we have trust by association. So when someone else I trusts you, I can also get in the door. So there's the thing that many people have been referring to. I've been at an academic medical center for 20 years now, and I can say that these are not insignificant. The deep culture of academic medicine that relies on our whole promotion system relies on individual credit for publications and grants awarded. And so this is in deep competition right now with the things that we know make research work faster and better, which is more open sharing. And so the idea that I think the thing that's promising and hopeful here is that there's not just one culture in science. I think that old model in academic medicine it's crumbling at the edges. We can see it. It's not going to last. And so, it is us. So it's up to us to really transform it and change it. And we have lots of successful models around us to help us build into this new space with academic medicine. So whereas competition, it's not taking competition off the table. We can look to open science spaces to see how these are incredibly competitive environments and spaces, but the competition happens in open space. And it's that open space that actually accelerates and fuels the competition. So there are ways, as we've seen in a couple of the examples already today, that we can do rewards, acknowledgement, attribution, and development, accelerate development, in an open space. There's no need to hold this so closely to our chest. We have, as I said, in many industries, we're seeing evidence of this change. So in information technology, we've seen this happen within our very recent lifetimes. So we went from kind of an expert-driven model where you had to have whole IT departments and massive server farms to manage your data access and data use. And now we're in this really user-driven, we've democratized data and data access and how people are using it in mobile and cloud source technologies. And we've really, in some really exciting ways, have made this available to everyone rather than the limited expert-driven few. So I think we can take, this culture shift has happened already in other fields, so let's move it into health research. One thing that can help us do that is staying grounded in our ethics. So these are the old-fashioned research ethics from the Belmont Report and many millennia prior to that. that really helped keep us in medicine and science remember what's important, which was respect, benefit, and justice. And if we think kind of with a new lens on these questions, so what does respect look like in this new open space? What does respect look like? How can we assure that our research processes are accomplishing benefits, and benefits for whom? Who decides what counts as a benefit? And have we created systems that are really equitable and fair? And if we for me, these are truly a touchstone. If we can keep coming back to these ethical foundations as a guide to us as we develop our practices and our processes for moving forward, I think we'll be on the right track. So just as a quick example of what respect looks like, I think how the Belmont Report got operationalized in our research ethics
Welcome to WebEx. Please enter your access code or meeting number followed by the pound sign.
Telling people what we were going to do and asking their permission to do that. I think there's a whole, that's one way we can demonstrate respect. There's a whole host of ways we can
Please enter your attendee ID number followed by the pound sign.
Think about it, respect has
if you do not know your attendee number, just press pound to continue.
So this is respect.
Welcome to WebEx. You will now be placed into the conference. You are the first attendee to join the conference. Please stand by.
So respect can be reflected through greater acknowledgement, greater expressions of gratitude. I'm taking an informal poll of researchers I work with on how often they just say thank you to the participants that are in their studies, and it's embarrassingly low numbers. So simply saying thank you can go such a long way. Having more exchange or more reciprocity in what is in it for me to enter into this collaboration with you. And some of the discussion we had after the last panel, I think, really pointed to that. Sometimes what might make a difference to someone in that partnership isn't what you think is valuable, but it actually is what's valuable to that person, and that's the thing we have to pay attention to. So amplifying other speakers, communication is just essential here, and really keeping a network going. So one way I've been thinking about this is the relationship between ethics and the law is the regulations really just provide the floor of our behavior. We can't go lower than that. But if we take the airline industry as an example, we want to do more than just what the regulations mandate that we do. Data sharing policies and such will start to provide a scaffolding for us, but it really is up to us as a community to set our standards of excellence and what's important for us. I really take a page from advocacy-run research groups in this. They've taught me a lot working with Sharon Terry and Genetic Alliance and PXE as a model of how there were 4 things that came out of some of our work together in building better biobanks that I just wasn't seeing as present in academic medical centers. So what I was seeing as different from an ethical perspective with the advocacy-run banks, research practices, success was right at the forefront of what they were trying to accomplish. They never lost sight of why they were trying to approach the research. Why were they trying to get the research done? It was for the health of people, and they worked with the relationships, the networks that they had. They forged new relationships, surprising alliances that they hadn't normally expected. And I think some of the alliances we're seeing in this room are one reflection of just that kind of creativity. I think a third principle was the idea of flexibility and being nimble. So when barriers did come up and get in the way, they didn't stop the train. They actually just found a way to move the tracks around that barrier and keep going. So it was keeping an eye on success throughout the whole thing. So just in closing, I'm hearing a lot of hope in this room, and that's just so encouraging. When I prepared the slides, I was thinking I might need to help provide hope, and Seamus Haney really does that, which is we've got a lot of history on our side that says don't hope on this side of the grave. But then once in a lifetime, the longed-for tidal wave of justice can rise up, and hope and history rhyme. And I'm hoping what we see here today is a lot of practical examples of how we can accomplish this together. Thank you.
Okay, good. Our next speaker is Dr. Ioannidis from Stanford. Can you hear us?
I can hear you now.
Okay, we're ready for you. Thank you.
Hear me?
Yes, very well.
Thank you for the very kind invitation. It's a great pleasure to participate in this very interesting workshop. I just want to make sure that the slides are being shared at the moment.
Yes, they are.
Okay. So in trying to describe my experiences on data sharing for clinical research, the best analogy that I could think of is, let's see. I don't think that the slides are moving, though. This is interesting.
Okay, they're on your, what is that, live meeting or WebEx?
So, okay, I think now it's working. So the best analogy that I could think of is trying to save books from a burning library. The ancient Greek world collapsed because, to a large extent, one of the main reasons was that it lost control and its scientific records. And the Library of Alexandria had about 1 million volumes at its peak. It was burned several times, and now we only have a few fragments and pieces that have survived out of that wealth of scientific knowledge. Currently, we are producing information and data that are equal to several libraries of Alexandria almost on a daily basis. However, most of these data are just presented as a synopsis, an advertisement, I would say, or even an editorial, which is scientific papers in a journal, a few pages that summarize sometimes petabytes of information. So how can we regain that information before it's too late? I have tried to use and retrieve clinical research data and participate in projects that involve data sharing for different purposes. These would be meta-analysis of individual participant data from randomized trials for primary or secondary outcomes on efficacy or harms, exploratory discovery analysis based on existing data such as biomarkers, development and validation of predictive models, methodological work, for example, risk modeling, repeatability and reproducibility checks, and also prospective meta-analysis and networks. Some basic principles is that it's more difficult or sometimes even impossible to unearth the data after the fact, after they have been collected, after the respective paper that describes them, summarizes them, makes an announcement or advertisement about them is published. So I think it's far better to arrange upfront for the full individual level data to be available, and then issues like coding, cleaning, logical queries that sometimes are considered to be a problem, they may take some time, but their difficulty is probably overrated. There is some difficulty of standardization in post hoc efforts that may limit the utility of some of the information, but if we think ahead of time of what exactly we want to collect and what exactly we want to share, this would be less of a problem. Some fields have used principles of data sharing routinely. So, for example, I think one of the major progress, one of the fields that had the greatest progress in the last 10 years has been human genomics, and the reason for that is that there are principles on how to share information at a minimum between all the investigators working on the same theme, and sometimes even more widely with other investigators or making them widely publicly available. I think if we did not have that possibility and that mechanism in place, we would never have materialized the fantastic growth that we've seen in human genome epidemiology or other fields in genomics. I think that clinical trials could learn from such paradigms and try to adopt them. If we take a look at the most highly cited journals, the 50 journals with highest impact factor in the scientific literature publishing primary data on what type of policies they have in place, this is an empirical evaluation that we published last year And we try to see whether these journals have policies in place for sharing different types of data, whether they have policies for provision of materials and methods and protocols, because otherwise, if you don't really have exactly a sense of what has happened and what the protocol was, it's very difficult to repeat any analysis or make sense of these data sets. And also whether there was a condition of publication that these data should be available. And basically that slide shows lots of color. If you see green color, that's good news. It means that most of these journals have policies in place for sharing data, for making protocols available, and even making a precondition of publication, the availability of that information. However, if you take a look at the last column that has these tiny font numbers, most of these numbers are zeros. And 0 means that 0 out of the 10 papers that we sampled from each one of these journals to see whether the policies were fulfilled actually had activated these policies. So even though there's a lot of interest and a lot of investment in trying to make data sharing to work, in practice I think that we still have some way to go. One of the most interesting aspects of data sharing is trying to see whether one could repeat analysis that have been already done. One doesn't necessarily have to come up with some brilliant new ideas to analyze. Occasionally, just trying to find out whether one could repeat the analysis that has already been published could be very useful and could give us a sense of how well the data sets are functioning if independent scientists are trying to do the same process. And we were very lucky that Nature Genetics and Miles Saxton were looking into the future a few years ago when we decided to do that project. And Nature Genetics, for many years now, has a policy that for microarray types of data, there should be a precondition to publication, a prerequisite to publication, that the information should be available in individual level information, and also the protocol should be available, and everything should be available to independent investigators. So we try to see whether one of the top journals with the best scientists publishing there, some of the best possible studies, whether they could be repeated and reproduced. And practically, 4 independent teams of microarray analysts could reproduce, in principle, only 2 of the 18 tables and figures from different papers that we had probed at that time. Much of the time, the problem was that the information was not available. So even under relatively ideal circumstances, still the data were not available to be used. Some additional problems that we have encountered in efforts to perform analysis with individual level information include politics. Sometimes people may perceive that this is private information that is boosting their personal scientific career, therefore it's not to be shared because other people will get that same competitive advantage. But I think that there's very little rationale for that. Actually, data sharing would only improve the visibility, credibility, and impact of some research. There's sometimes a clash of evidence-based medicine with expert-based medicine. Both of them are EBM. I think that expert-based medicine remains extremely more powerful compared to evidence-based medicine. Even nowadays, I have a paper with Peter Getze coming out shortly in BMJ on that, on what do you do with involving experts in evidence-based projects, that some of them may involve sharing individual level data. Another issue is that not all data sets may be available at the individual participant level, which means that this generates new possibilities for selective reporting biases, and that could be a serious problem if, for example, there's 50 randomized trials that have been performed on a topic and only 20 of them could be retrieved in individual level information, then maybe these 20 are a very select sample. Maybe it's only people who believe in a particular type of treatment effect and result that would share their information, and therefore the meta-analysis of these individual level data would be less credible compared to meta-analysis of just summary published information. And I think that all of these problems are largely almost eliminated if sharing of the individual level information is agreed before the data is generated. I think if we wait to see what we can do after the fact, it's very difficult. Finally, I'm showing a slide that seems to be very convoluted, but I think that this is probably what happens in most fields of clinical trial research. The leading paradigm has been that a clinical trial is an island on its own. It can be designed, conducted, analyzed, and published, and we get an answer, and this is it. Most of the time, this is not the case. Usually there's a universe of trials. There's many trials that each one of them may inform the results of the other. And even if you take, for example, neglected tropical diseases by their name, by default one would think that these are diseases that no one is doing research on. Actually, there's tons of trials. This is from a paper that will be coming out in BMJ shortly, where with Shanti Kapagoda, I have tried to map the geometry of the networks of trials in neglected tropical diseases, and you see that they're extremely complex. There's all types of comparisons that have been envisioned, and if someone is trying to make sense out of the results of a single trial without having the ability to juxtapose the results of all these other trials and analyzing them with some standardized fashion, it's very difficult to find a final answer. So I think that my reply to the question, do we need clinical data sharing, would be yes, and probably with no qualifications and caveats whatsoever. Thank you.
Thank you very much, and thank you for keeping to time. Can I ask the other panelists to come up? We have about 10 minutes, I think, to have a brief conversation, and let me introduce Sally Oken from Patients Like Me, and maybe you can begin with short remarks, and if you don't mind giving us back a little of the time.
I don't mind at all. I appreciate being here and the opportunity to be able to address some of the things that came up this morning. I think we have some common themes. I'm from Patients Like Me, and for those of you who are unfamiliar with it, Patients Like Me is a research-based, clinically relevant site for patients, peer-to-peer relationships where they can establish profiles, provide health data into the system, share it amongst each other and amongst us as researchers, and then we make that data available in an aggregated way in ways that we de-identify it and then try to make some sense of it for people who might be interested in that. That's our business model. The site is free for patients. There's no advertising at all. So I was asked to give a little bit about what of the lessons that we've learned. We've been doing this for 7 years. I think we're innovators in really gathering patient-reported data. We are not doing clinical trials, so I want to say that quite clearly. However, we are doing research. So one of the things I want to start with is one of the mandates that we say to ourselves is when someone gives us something, we need to give them something back. So it's our give something, get something motto. And I think that can be looked at in a variety of ways. One way is that when we've actually started downloading all the clinical trial .gov data every night, and every morning patients can go onto the site with one click, see the data that they've given us. We will then show them all of the trials that they're eligible for based on location, by age, by gender. So it helps to simplify the process. But if you think about it, if patients have given you data for a research project, then you should be giving that back to them in some fashion. That's identifiable for them, that they can make some useful, meaningful use of in terms of their own life and decision making. The next is patients first. We feel very strongly that when we are engaging patients in a data sharing environment, that we need to put their interests first, and we need to understand that they have a lot more skin in this game than any of us in this room do. That they have about 90% of the time spending more of their time doing self-management of their care, so they are the experts in their condition. They have the expert knowledge and the expertise in managing it that as clinicians and as researchers we could just never have. We need to hear that. We need to listen to that. We need to collect it. We need to understand it. Then finally, listen well. I think this is probably one of the most important things to me. Patients have things to say, and frankly, they want to tell us stuff. We've recently participated in an IOM discussion paper on effective communication. It was just released about 2 weeks ago. And one of the questions in that we have actually expanded on and are now doing a full survey on was asking patients, how do they feel about the statement, my health data should be used to help improve the care of future patients who might have my same condition. Overwhelmingly, 89% of the people who were in that survey, there were over 1,000. It was a general population of patients, so it wasn't people in the population. It wasn't patient necessarily. 40% of them all reported at least one condition. 89% of them said, yes, I feel very strongly that my data should be shared for that purpose. Yet, when asked whether they thought it was being shared, the majority said they either didn't know or no. So we have a huge gap between what patients are telling us they want and what they perceive us to be doing. So finally, I think I want to make a couple of provocative statements, and I'm going to give you the time back. One is when we listen to patients, we need to be recognizing that the data they're giving us is intimate data. It's data about their daily lives and the things that they're dealing with every day. They are not simply human subjects. They are participants who are actually members of the research team. I would change our paradigm completely and start thinking of patients as patient researchers or citizen researchers, however we want to call that. I would also say that we have to start capturing the patient vocabulary in ways that it's better understood by clinicians and researchers. We do that at Patients Like Me. Every piece of data that comes into me, we curate it, we keep the patient voice, and we curate that against a coded, clinically known terminology. So you'll hear the patient voice first, and then you have to look and see what the clinician result of that might look like. And then finally, I think we need to really recognize that when you empower patients and then you engage with them, they will activate if you give them the right tools to do it. And there's been a recent blog post that said if patient engagement were is really the blockbuster drug of the century. And if that were the case, if it were a drug and we weren't using it, we would be considered practicing malpractice. We would be providing harm because we're not using it. So finally, the system's never going to be perfect, but I think we are at a place today where we're starting to talk concretely about all the stakeholders, and I'm going to just constantly bring us back to the fact that without patients, we would have no research. So let's start thinking about how we can best honor them, respect them, and allow them to develop the trust that they need to trust to participate with us to find the answers so that they and their clinicians can start solving the problems of their medical conditions. Thank you very much. You've got your time back.
Thank you so much. I think that was exactly what we needed to have the patient's perspective. Please take my chair. First question that came in, we only have a few minutes for questions, I think is right online. You've heard the tension a little bit between the stakeholders of you, and one of them really centers around privacy versus productivity, we heard earlier. And the question that came in is how much will the issues of cost and time and the impact the ability to de-identify data? And I think Richard was the one who presented that de-identification was a major project in the Yoda experience. Maybe you could start.
Well, we're pioneering this. I think there's nobody. I think we're the first industry group to actually give this patient-level data out independently before. So it clearly is a Rube Goldberg contraption right now to try to figure this out. And what we did was not scalable. That's for sure. And I think what we wanted to do was comply with HIPAA. HIPAA was designed not for this activity, so trying to fit that square peg in the round hole was probably why it cost so much and we're moving around. So I think that the theme that we were all talking about here is as we look at all the different dynamics going on now, the way data is collected, digital medicine, the movement from proprietary data to sharing data requires a lot of other foundations to change as well. And I think that we probably should rewrite the notions about how HIPAA works with an eye towards facilitating this process quicker and also get the patient's voice in there. And I think, as Sally had mentioned, we might find that some of those barriers to identification may be laxed to some degree when patients are participating as partners rather than subjects.
Any others?
Although the amount of time and money it costs Medtronic and others in terms of the models we've heard sounds high, and of course it is high, and it might not be scalable. I think it's really important to put in the context of the profits or the earnings in the case of industry for the product at hand, or just the amount of money that's spent on the clinical trial, if that's what you're talking about to begin with. So the cost of disseminating the information from the clinical trial at least needs to be put into the context of how much time and effort you put into the trial.
Yeah, I have to say, I mean, our experience with DataSphere has been slightly more modest in terms of time. We logged this very carefully with the biostatisticians that we were using, and it took around a week to 2 weeks for the two major data sets of programming time. And we spent a lot of time with biostatisticians and so on, but we have the 17 key areas from HIPAA, and then we have this modified statistician method. So there's a little bit of a variance in terms of the amount, but I think I agree with you, Deborah, when you're looking at sort of $100 million data sets, which some of these are, the value that's there, it's well worth investing the time in 2 weeks of biostatistician and programming time to make these data sets available.
Thank you. Why don't we take a question from the audience, and I'll pose one before that if there's someone who'd also like to share one. One question I have, and this was sort of brought up by at least one of the speakers, is the time and complexity associated with assembling these data sets and partnering with academic groups, making contracts across different companies, etc., may be actually a burden and a barrier to the actual successful assembly of the data that we all say should be published as a public good. So one question I'd ask for this audience, recognizing there are no members of the regulatory agencies on the panel, is what is your view of the transparency of the regulatory activities and whether or not regulatory agencies should be part of this process in analyzing, transmitting, and exposing public data, recognizing that under the current guidelines it's not permitted, but they are, of course, discussing it in the EU, as many of you know.
Jeff, I wanted to speak to this point because I dealt hands-on with the complexities of putting the data sets together, and I just wanted to mention that the FDA has an initiative right now, and I'm part of a group of people who they solicited their opinions about uniform data submission, required data fields, and how data should be collected for trials. And this could facilitate these kind of efforts in the future.
Just to add to that, CMS, which you probably don't think of as an agency that does research, CMS has just launched this summer an office dedicated to pushing out CMS claims data to researchers and other recipients. The director has just been named. I think this is in part coming out of health reform, where if you're going to add a lot of new folks to the insurance rolls and control costs, you have to control the cost of the care that each additional person gets. And there's, I think, some insight there that all of this existing claims data tells us something, as you were saying, the care that the patients are getting is valuable. So I do think you're starting to see a change at the government level.
Any other comments? All right, let me, please, thank you.
Issues, and now the other issues related to patients. Patients seem to be sort of the barrier in here in a lot of ways because a lot of the protections are for them. But let's just go back to what we learned in kindergarten, because I'm a patient and this is all about communication. We need to say please, we need to say thank you, and we need to just ask people to share. And everybody gets a trophy because that's the way they play t-ball now. So if we just look at those principles and move forward and not just keep making the same mistakes, then I think we have a solution, and I thank you all for being here.
Comments, please.
Just a quick comment to appreciate what you're saying about asking, because I think HIPAA is there, and we're spending a lot of money and time on removing these HIPAA identifiers, but that's a regulatory fix that was trying to protect people. But there is another way to think about privacy, and that's the positive right to privacy, which is I control who has access to my data. Not just my data will be hidden and anonymized by someone else, but I control who has access to my data. So if we ask someone permission, as patients like me has seen, they might choose to put a lot of identifying information in there. So I think you're right on target. We should do more asking.
Yeah, Cindy, I agree with you completely. I mean, we've spent a lot of time looking at legal issues, IT issues, clinical issues, biostatistical issues. And while they're all complex, I think the single biggest thing that prevents us with data sharing is effective communication. We see it as about 80 or 90% of our project. All the other things are sort of more mechanical processes you can get through. But dealing with people's emotions and communication is one of the biggest challenges we face. I agree.
We're running a little bit over time, and I'd like to get everyone to lunch now. And so I just want to check, Rebecca, what time should we be back? 1:30. Okay, so we only have about 20 minutes, 22 minutes. Thank you very much for an excellent panel and discussion.